Sundar. He is a data scientist and advisor-in-chief at BioFlues. Please, sir. I would like to invite this session with the announcement of the Bird Award by the BioFlues. This year's Bird Award is going to Dr. Manjari Kiran. I congratulate her. Dr. Dr. Manjari Kiran is an assistant professor at the Central University of Hyderabad to encourage bioinformatics in India who have made outstanding contribution in the area of bioinformatics and functional genomics. We have initiated the BIRD Award in 2011. This award is meant not for just academicians but also scientists who have shown exemplary excellence in the field of bioinformatics. While scientists who have been working for not-for-profit organizations will be given importance, the ones who are working for commercial organizations are no less exceptional. If the research they have carried out have been widely honored and have made long-standing contribution. Dr. Manjari is an exceptional candidate. As an awardee, she should receive a plaque with certificate. I invite you, Dr. Manjari, over the stage.
Good afternoon, everyone. I'm deeply honored to receive this award, and I, uh, I, I hope this award will keep motivating me to do my part, to contribute advancing bioinformatics in India, either by training young minds or by doing research work I do. So, today, I will be showing you what we are doing in our lab, which is in University of Hyderabad. Stop. Do we need this? Hello, hello. Am I audible? Okay, good. Okay, so many things to hold. Okay, thank you. Yeah. So this. First of all, thank you for inviting me and giving me opportunity to talk, present my work here. And this is special because of couple of reasons, very important reason. One is that Banaras Hill University is my alma mater. I have done my bachelor's and master's from Mahila Vidyalaya, MMV, what you see when you enter BHU. So when I got to know that this event will happen in BHU, I could not resist. I wanted to see this place again, and I am coming here after 16 years. So everything feels like still everything is same, and I wish to visit each and every department which I visited that time. That is one reason. Second reason is whomever I have followed throughout my academic career, whether it is Professor Lokotia, Professor Raghavan, I am seeing them in the audience, and it makes me like I'm excited for that. Also makes me nervous that I have learned from them by following the research altogether. So thank you once again. So in today's talk, I thought of giving a very generic title, which is Interpretable Machine Learning Models for Biological Problems. I apologize for the font. It was different on my laptop. It became different here. So why I'm saying interpretable? Because we do a lot of machine learning, but from this machine learning, we want to learn from the model that what are the determinant features and how these features will help us to understand the biological problem. Okay, yeah. So my lab at University of Hyderabad in Department of Systems and Computational Biology majorly focus in two areas, RNA biology and genomics. Last couple of years we have worked on RNA biology. I'm very much fascinated with non-coding RNAs, whether it is small non-coding RNAs like tRNA-derived fragments or long non-coding RNA itself. So we asked the question that can we determine some factors which can help us to calculate number of alternative splice form RNA can have, and if we can do so, can we predict number of alternative splicing forms by given number of exons? And this idea came up with the observation that whenever I work with non-encoding RNA, which are RNA of more than 300 nucleotide bases, I always notice they have lot many transcripts, although they are low expressed, but they have many transcripts. So how to find, like, get a estimate that given number of exon, how many alternative splice form a gene can have? So for that, one of the master student, Kaushiki Basu, very talented student, she worked on it. And first things what she found that coding potential and exon length plays an important role. And because coding potential plays an important role, we notice that non-coding RNA which have very low coding potential, they tend to make more number of alternative splice form. And this is also facilitated by a different 5' splice site regional link RNA. And this all together actually is because of the inefficient splicing of the non-coding RNA. So the whole data is present in this paper if you are interested in. And we have also developed a, excuse me, linear regression based model to calculate this. Sorry for the disturbance. Yeah. Thank you. So this paper presents a method to calculate number of splicing forms. Yeah, I have water. Thank you. Yeah, I have a little bit cough, so excuse me for that. Yeah, for cancer genomics, uh, what we are starting working with is the genetic interactions. There are different types of genetic interaction in cancer, and we're trying to find features that can classify this type of genetic interaction. And once we find these features, we want to see whether we can predict different types of genetic interaction with one multi-class model. And this work is done by Anwade, which is recently published in PNAS Computational Biology. Thank you very much for this. I was asked to present for 15 minutes. Now I'm taking more time. Okay. So in terms of RNA biology, we are moving towards whether link RNA can encode microproteins, and if they can do, what are the RNA modifications involved in it? So more like epitranscriptomics. And in cancer genomics, we are moving forward to single cell RNA sequencing, and how we can use single cell RNA sequencing to identify genetic interaction. What are genetic interactions? They are phenotypic outcome resulting from two or more gene interactions. So most of you probably know about synthetic lethality, which is a very common genetic interaction found in cancer. There were three independent groups which identified that part 1, which is a DNA repair enzyme known to repair the DNA breaks. Usually, if it is inhibited, there will be eventually a double strand break. In normal cell, it can be repaired by homologous recombination, but in BRCA mutated cell, homologous recombination cannot happen. The cell will undergo death. And in the usual scenario, it can be repaired. But there is one more genetic interaction, which is synthetic viability, which is one example I'm showing you here is between HDAC and DBL. Both are positive regulator of North signaling pathway. So it has been shown that if HDAC is inhibited, there is alteration in DVL, which leads to cell proliferation. So here the phenotype is cell death, and here the phenotype is cell viability. So this is called a synthetic lethality. Cancer cell would die, but patient would survive better. Good for patient, bad for cancer cell. This is where the cancer cell survives. Bad for cancer cell, good for bad for patients, and good for cancer cells. So there are different methods to identify genetic interaction. I am not going to details of it. There are different experimental approaches. All of these have their own drawbacks, limitations. Now, even if we go with CRISPR, we have information of around 4,000 such genes, and also these genes are more or less of interest of the lab. So there are computational approaches required in that usually the input informations are copy number variation, gene expression and mutation obtained from DCJ data. So there are statistical pipeline to identify genetic interaction. These models have their own limitation because they are based on lots of assumption, and if there are not enough number of power, which means if there are not enough number of samples, we fail to identify a good genetic interaction pair. So what I am going to talk about is machine learning methods, which we have used in our work. So among all the machine learning methods which are available to identify genetic interaction, most of these methods are binary classifier. So they predict whether an interaction is SL or not SL, whether the interaction is synthetic lethal or not lethal. They are trained on less number of data. They are trained on imbalanced data. They are non-interpretable. So taking all these limitations, we wanted to develop a multi-class classifier which can predict any interaction, any given interaction as synthetic lethal, synthetic viable, or not interacting. That was the aim. So for the feature, for any machine learning method, we need features, and for features, we took physical protein-protein interaction network and the network properties of each node. So what is a protein-protein interaction? So here I'm showing you a toy example of protein-protein interaction where each nodes represent a protein, and the size of nodes which I have simply to signify that these are important node, or in other words, central node of the network. So the questions what we are asking is that if you take any SL pair, which is synthetic lethal pair, how far are they located in the network, or how well they are connected to other nodes of the network? That is one question. Same for SV. So can we use the information of how these genes are, or how these proteins are placed in the protein-protein interaction network, a physical protein-protein interaction network, and use it as features to learn whether a pair is synthetic lethal or synthetic viable? And the whole rationale behind this is there were a couple of papers showing that the showing that the protein products of these genes, they do physically interact. That is one. Secondly, there was one more paper which showed that synthetic lethal pairs are very close in the network. And they talked only about synthetic lethal pairs. Nothing is known about synthetic viable pairs. So we wanted to learn that how these two genetic interactions with a completely different phenotype, they look like on the network. So for that, we have used biograde experimentally validated protein-protein interaction network. And some of us may be interested in the proteins for which we do not have experimentally known partners, so we moved to string as well. So we have two models, one for experimentally validated ones, another one for computational plus experimentally validated ones. And for the properties, we have used node-wise property and pair-wise property. So in node-wise property, link degree, which is number of interaction a node has, betweenness, how many shortest path pass through a node. For example, this node, any talk between this module to this module will happen through this node, so it will have high betweenness. Neighborhood, how many partners are overlapping. Triangle, how many partners are talking to each other. And they are pairwise property, which is between two genes, how many, like suppose if these two magenta genes, how many paths you need to take to reach to that gene, shortest path you need to take to reach that gene. Like that, there are pairwise properties. So as I said, questions were how are SN has been placed in the physical protein-protein interaction network, and how different are these pairs in terms of network properties. So for that, we developed a multi-class model which we called Magical, and we have taken experimentally known synthetic lethal and synthetic viable pair from these three databases. So all these interactions are known by RNAi or CRISPR. And as you can see, there is a huge imbalance in the data. So we took care of the imbalance by using SMOT algorithm, which not just undersample one of the majority class, but also At the same time, oversamples the minority class. So it does take care of both the classes pretty well. And the major hurdle in front of us was to create NOT data, to actually find the gene interaction which do not ever, they should not genetically interact. So how do you call something not interacting? That's hard to do. But what best we could do was we took all the proteins, made possible pairs, remove all the known experimentally known interaction, and also the ones which are known in yeast, the homologue of these in human, and what we were left with not. There were lots of knots. So every time we picked 10,000 knots so that the number looks similar. Now the training will be totally dependent on what knot you are picking. So that's why we did 1,000 type bootstrapping. So every time different knot set was picked. So every time balancing the data and knot was done. So altogether 1,000 models. So once we have SLSB and knot, we mapped it to the protein-protein interaction network, and we calculated all the network properties. We found the discriminatory feature, which became our tool, and then we saw whether it can predict SLSB or not. And the result from 1,000 models showed that most of the models, they gave us accuracy which is 24%. And among all the 20 network properties, shortest path was picked by almost all the 1,000 model at the first rank. So all these 5 properties, what you see here, shortest path, average neighborhood, average betweenness, triangle, and adhesion, these were the ones which were discriminatory enough to call something as SLSB or not. The same is shown in the graph here, that SL genes, the genes which are involved in synthetic lethality, they are placed far in the network. SBs are close. So if you think of any process of drug resistance, like one gene is inhibited, the other gene which alters and rescues the phenotype is a gene sitting next to it in the network, actually. Okay. So SLs are far. They are not just far, they also are, adhesion also tells the same thing, that they are far in the network. It's very easy to separate them to two subgraphs. SVs are close, and SLs are very central in the network, which means that they are important protein, and of course they should be, because many of these are cancer drivers. We tested it in test data set, and in both magical core, which is experimentally derived PPI network based training, and string data set, we see a good accuracy. Magical worked really well with independent data sets also, like we checked it on depth map data, which is a high throughput screen, CRISPR screen data set, and also on publicly available CRISPR data, and we saw that Magical is able to predict pretty well. Comparing with the Bannet classifier, which was available based on network properties, we see that Magical is predicting better than SLANT. And based on functional models, what we found that SL pairs share less functions compared to SV pairs. So these genes, which are supposed to functionally should be redundant, that if one gene is inhibited, the other gene takes care of it, they don't share many functions. They are central in the network, they have many functions linked to it. They have very limited functions which are overlapping. That was something new what we found in our work. So this is the inference, where we showed that SLs are part of different network, different sub-network, different communities. SVs are part of same communities. Functionally, they are more unrelated, but SVs are in same pathways. And this is specific for glioblastoma. What I showed till now was pan cancer. For glioblastoma, I'm showing you one example where these two genes, RECQL is a ligase, TP53 is tumor suppressor. If the patients are harboring mutation in these two genes, the survival of the patients are better than the ones which are not having any mutations, any alteration. This is SV pair predicted by Magical, which shows that if there is an alteration in BRAF and PTEN, BRAF being oncogene and PTEN being tumor suppressor, the survival is poorer compared to the ones where the status is wild type. And this is already reported in the literature. We also have a database where we have hosted all these interactions. This is last slide. Yeah, so one of the pair is already in the clinic, not from our lab prediction, but when we cross-checked, we found that one of the pair between BRCA1 and ESP1, Magical is giving it a score of 84%, and it's already in a clinical trial. And this is my lab, and we have funding from this. Thank you. Any question, please?
So, do you have any restriction to number of nodes in each network? In a range, you set up like minimum or something?
Nodes are all the proteins which are in human proteome. So there is no restriction as such. So we have one experimentally derived physical protein interaction where we had around 14,000 nodes, and in computationally predicted ones combined with experimental, we are almost covering all the proteins.
So how do you keep annotation updated with these pathways in network? Because annotation is keep evolving, right?
Mhm.
So how do you keep updated?
So for the training of the model, we don't need any annotation. So for the training of the model, what information we need is where are the nodes placed, genes placed in the physical protein interaction network.
It's unknown.
Yeah, yeah, these are known, so that doesn't change much. Yeah, thank you.
Hello.
Any other question?
Yeah. Sorry, from this side.
Yeah, please.
Model expectation and referring to spice level.
Uh-huh.
You have expected some model, your model.
Mm-hmm.
Is it matching with reality? Naturally, slice variant.
So you are talking about the RNA biology part.
Yeah.
Yeah, we did check for a couple of examples. We were also asked us to check that, and we came up with linear regression. We came up with a formula. I did not show the formula here. If we know the exon length and know the how many histone marks it has, histone acetylation marks, we can predict how many alternative splice form that gene can form, and it does match to the real data.
Natural data.
Yes.
Is it across species or?
Or a human and mouse.
Thank you.
Okay, thank you, Dr. Manjari. If any further questions, is available here only. So one can interact with outside now.
Thank you.
We are now pleasure to introduce Professor G.K. Raghava, who is a distinguished professor at IIT Delhi, and he also got his MTech from IIT Delhi and PhD from IMTech Chandigarh, and he worked as postdoctoral fellow at Oxford University, and he is a specialist in UAMS and also visiting professor in Hosted, South Korea. He worked more than 30 years and is got his lab 350, more than 350 now, research papers in reputed journals. Some of the things developed more than 300 in silico products like web server, databases, and software packages. And some of them were the highest contribution in the world from any single group. So we are very honored to have here Raghava today for having this. We welcome. Somebody else?
Okay, so I would like to thank Dr. Manjari for her talk. It was amazing and enlightening, and I thank her for sharing her expertise on interactions and their role in predicting the diseases. And now I would like to invite Professor Lohia to facilitate her with the burden of. Please, sir. Thank you so much, sir, and thank you so much, ma'am. And now I would like to hand over the mic to our session chairs.